it has been shown to implicate negatively in cardiovascular disease, including atherosclerosis. Oxalate has also an increased effect in inflammatory disease, including inflammatory bowel disease. When we talk about inflammation, one of the important players upstream of many inflammatory pathways is myeloid differentiating primary response protein 88 or MyD88. MyD88 gene produces an adaptive protein, which is very important for signal transduction, and it signal transduces using the toll-like receptor family and is important in innate immunity. Studies have shown that MyD88 is involved in numerous inflammatory responses, including its exposure to oxalate. It also regulates tight junction permeability in renal and intestinal tissues. Research has shown that when you delete MyD88 in an intestinal specific manner, it partially protects mice against inflammation and renal injury. So we used a germ-free intestinal epithelial cell-specific MyD88 knockout and the legitimate wild-type MyD88 wild-type as control to elucidate the mechanism linking oxalate exposure, hyperoxaluria, and colitis. So our goal was to see, when you feed mice a high oxalate diet, what happens to these various aspects? How does it adverse the mice negatively? So in a germ-free facility, we derived these MyD88 wild type and knockout. And the MyD88 was only deleted in the intestinal epithelial cells. And we had to give tamoxifen to induce the knockout. And after we had the knockout, we used the wild type as legitimate controls, and we fed them 1.5% oxalate diet for 14 days. And alongside, we had a basal diet to compare. Sorry, this slide usually takes a little long to come up because of the image. Here we go. So we looked at how oxalate affects the colitis in mice. And we looked into the crypt density, crypt hyperplasia. We observed the goblet cell loss, the muscle thickening, the amount of infiltrates. And the colitis scoring was done by 2 independent investigators. And what we saw was, in both the knockout and wild type, oxalate was able to induce colitis-like phenotype. So basically, oxalate-induced colitis in these MyD88 wild type and knockout mice. However, again, this slide will also take a second to come up. Yep. What we noted was, compared to our MyD88 wild type, which is here, the knockout had lower colitis phenotype, suggesting that our MyD88 knockout model was slightly protective to the oxalate-induced colitis. However, the fact remains that oxalate in the diet increased the chances of these mice having colitis. Can microbes come to the rescue? Because we know traditionally that oxalate is degraded in the gut. So to understand that, we went back to the microbiome of white-throated wood rat, or Neotoma albigula. It consumes a diet comprised of oxalate, and they are found in the deserts of Utah. However, they are capable of degrading a large amount of oxalate without having any adverse effect to the system. When we looked into their microbiome using metagenomic analysis, we found that about 59.27% of all the genomes contained at least one oxalate-degrading genes. Our next question was, when you degrade oxalate, you get formate and carbon dioxide, right? What happens to those byproducts? And when we looked at the metagenomic analysis of the microbes important for the formate metabolizing genes, methanogenic, acidogenic, we found that they are almost universal. And the sulfate-reducing genes were found in 31.5% of all genomes. So basically, it's to say that, first of all, when oxalate is degraded by traditionally Oxalobacter formigenes, it forms carbon dioxide and formate, which can be used by these acidogenic, methanogenic, sulfate-reducing bacteria and formate metabolizing bacteria to give rise to acetate, methane, and hydrogen sulfide. which again can be used by the commensals to be utilized in citric acid cycle, gluconeogenesis, and fatty acid synthesis. So basically, when oxalate is degraded, the byproducts are utilized by other microbes, and then they are further degraded to make a whole cycle. Based on this, we can think about 3 different hypotheses. One is the traditionally known hypothesis that one microbe, one function. Oxalobacter formigenes is solely responsible for oxalate degradation. But studies have shown, clinical trials have shown that not everyone responds to Oxalobacter formigenes as a probiotic. So the second hypothesis would be there are multiple microbes with many functions, which is the cooperative function that I just spoke about. Or the third alternate hypothesis is there are multiple microbes who are capable of degrading oxalate, stating one function, or redundancy. So to test this, we use different microbes taken from different oxalate diet trials, and they included various groups. They had we had oxalate degrading bacteria, we had acidogen, methanogen, sulfate reducing, and we had some commensals in the mix. And these microbes were obtained from ADCC. First, what we did was some in vitro analysis. Here we used a media rich in growth, it had a growth media and it also had oxalate, and we tested to see if these different microbes could degrade oxalate over a period of 5, 14, and 21 days. And what we found that most of the species were able to degrade oxalate, suggesting that yes, in the gut, apart from Oxalobacter formigenes, there is a possibility multiple microbes are doing the same function. Now the question is it cooperation or is it redundancy? Meaning, is it one function or are they producing many functions? To test that first, we looked into how oxalate was degraded in an in vitro phenomenon. And to do this set of experiment, we subdivided our microbes into different groups. First would be just the blank media, which had only oxalate, no microbes. Second group had oxalate degrading bacteria or Oxalobacter formigenes. Third group had the oxalate degrading bacteria plus the acidogenic methanogenic sulfate reducing or the format utilizers. The fourth group had all our microbes, and the fifth group had everything without Oxalobacter formigenes to test out how they would act when Oxalobacter formigenes is absent. We saw in in vitro results that, as expected, Oxalobacter formigenes degraded a large amount of oxalate at 5 and 14 days. However, the rest of the group, including the group that did not contain Oxalobacter formigenes, were capable of degrading a significant amount of oxalate, suggesting redundancy. Now, what happens to the byproduct of oxalate degradation, which is formate? We tested that, and these bars represent Oxalobacter formigenes. And as expected, we see that there is a high amount of formate released in the system. If you notice the rest of the 3 bars, specifically in these 2, this one containing all the microbes and this one everything without the Oxalobacter formigenes, we notice that there is an increased amount of formate forming at the 5 days, suggesting oxalate degradation. However, at the 14-day time point, there is a decrease in formate amount, suggesting that the microbes are utilizing the released formate, which points towards our hypothesis of cooperation. So to summarize the in vitro part of the data, we can say that yes, multiple microbes can degrade oxalate, and oxalate and the formate metabolism is both redundant and cooperative. Now, how does this work in our germ-free system? We have these mice, we know they get colitis with oxalate. Can our groups of microbes mitigate the adverse effects of oxalate-induced colitis? Again, we had the same 5 groups. We had a germ-free control, meaning they only got PBS. Then one group got Oxalobacter formigenes, oral gavage. One got Oxalobacter formigenes plus the acidogenic, methanogenic, and sulfate-reducing microbe. One group got all the 8 microbes. And finally, a group getting everything without the Oxalobacter formigenes. And they were given 1.5% oxalate diet plus the basal diet. First, we see the amount of oxalate detected in stool increased both in knockout knockout and wild type after the oxalate diet in the group that did not have any intervention. However, if you notice both here and here, which represents the different groups of microbial garbage, the amount of oxalate detected in stool decreased with our microbial communities, suggesting that they are able to degrade oxalate sufficiently. Similar results was noted in the plasma oxalate, specifically in our wild type mice, where we see that initially there was an increase in our non-intervention group. However, the communities were able to lower the plasma oxalate detected. This is perhaps one of the most important slides where we wanted to check if the microorganisms were able to reduce oxalate-induced colitis in our germ-free mice. And if we see, this is the wild-type graph and this is the MyD88 knockout graph. We see there was an increase in colitis marker in both of them post-oxalate. However, different groups of microbes were able to mitigate or lower the oxalate markers in them. But the key takeaway here is the group containing all our microbes was the most efficient in lowering the oxalate-induced colitis, suggesting, again, a cooperative phenomena might be happening within the system, which is helping a group of microbes to mitigate the adverse effects of oxalate the most. To summarize the result, our oxalate diet did induce colitis in mice, and either the conditional knockout of MyD88 or our complete community mitigated these effects. So in conclusion, our study demonstrates the detrimental impact of oxalate in overall inflammation and colitis. When we targetedly knock out MyD88 in the intestinal epithelial cells, or our microbial inoculation rescues oxalate-induced colitis. So this indicates these microbes have a protective role. And we are working towards doing a clinical trial to use the set of microbes as a probiotic. And we also have a biomarker assay which is developing to detect which patients will actually respond to this or oxalobacter-found genes. I would like to thank all my lab members. Thank you, ASM, for giving me the opportunity. And with that, I'm happy.